Okay, this is a video lecture for Chemistry 255, Introduction to Biochemistry at St. Francis Xavier University. In this lecture, we're going to continue our module on proteins and specifically focus on primary and secondary structure of proteins. In previous lectures, we've talked about amino acids, how they function, their characteristics, and now we're going to look at how we put them together in a functional protein. When we left class last time, I left you with this problem, the titration of lysine with sodium hydroxide. I had showed you how to calculate the isoelectric point of an amino acid. That is the point where the amino acid has a net charge of 0. And left you with this example to do on your own. We were going to do it in class, but we won't have time to do that. So I strongly encourage you to try that out on your own still. We just won't have time to do it in class. So this simply highlights the transitions of a free amino acid. Transitions being changes in the characteristics with pH. And you can observe changes specifically in charge with amino acids over a range of different pHs. Because amino acids themselves are weak acids. Specifically, they have 3 groups that can ionize: the alpha carboxyl, the alpha amino, and usually the R groups. Some R groups can ionize. And so these will have a strong buffering effect on hydrogen ions in the body. So it's important to understand how these side chains, these functional groups ionize in order to figure out how protein shape and protein function will be affected. Now the isoelectric point is the point where the free amino acid has a net charge of 0. So some individual functional groups might have a charge, but the overall charge of the amino acid has a charge of 0. And to calculate the pI, you'll need to know that it's the average of the pKs around the point where the amino acid has a charge of 0. So if you figure out the net charges on the molecules, you can use that information to calculate the average of the pKs around the point where the amino acid has a charge of 0. Because remember, a pK value tells us where that form is in balance with its protonated and its deprotonated forms. So at a pK, there will be half of one form and half of the other, usually half of the protonated and half of the deprotonated forms. So the pK value itself does not tell you the point, the specific point where the amino acid has a net charge of 0. It tells you where that form is in balance between its 2 different forms. So work through that example and see if you can calculate the pI for lysine. Now keep in mind that all of this information is for a single amino acid. Not often do we find single amino acids on their own in the body. Usually they are linked together in a polypeptide. When that happens, the alpha amino group of one amino acid is bound to the alpha carboxyl group of a second amino acid through a peptide bond. And we've talked about what that bond is. We've shown the characteristics of that bond. And so as soon as that bond happens, as soon as those 2 ends bind together, those are now not free to ionize. That said, calculating the net charge on a polypeptide is easy. You have one alpha amino group that you need to observe the charge for. That's on the N terminal, the far left end, when we write it out in standard notation. One carboxyl group, the carboxyl group on the carboxyl terminal. And then you need to know the charges of any R groups in that polypeptide. So if you have 10 amino acids linked together or 100 amino acids linked together, still one amino, one carboxyl, and then the influence of any charged R groups will influence the total charge of that molecule. Now I want to take a second to talk about amino acids. We've set the stage with 20 standard amino acids, and that is indeed the case. There are 20 standard amino acids that are included in proteins, but some amino acids can be modified. By no means are there only 20 amino acids in the body. Some are slightly modified and some need to be modified in order to carry out their proper function. Now, modification will affect things like charge. It affects the characteristics, the solubility of an amino acid. And therefore, it can affect how a protein will shape itself, how it will interact with other proteins, and therefore it will affect its function. Now, some examples of modified amino acids are shown here. And in each case, you don't have to memorize the name because the name really describes what happens. Let's take, for instance, this O-phosphoserine. That simply says that a phosphate is attached to an oxygen on the R group. Normally, serine is an uncharged molecule. It's polar, but it's uncharged. Now it has a strong -2 charge with that phosphate group. So you can imagine that will strongly attract any positively charged amino acids within the same polypeptide or within another polypeptide. This carboxyglutamate example, normally glutamic acid has a -1 charge. Here we can see another carboxyl group has been added. It's strengthened this negative charge slightly. The same is true for these other amino acids that are slightly modified. The name tells you what happens. This 4-hydroxyproline we're going to come back to when we talk about collagen formation in the tertiary structure section. This is the amino acid that is critical for proper collagen formation. And it's this amino acid that is not formed properly without adequate vitamin C intake. When we don't have enough 4-hydroxyproline, this is when we observe the occurrence of scurvy, and I'll tell you how that happens. Now, some of these are modified and included in proteins. Some of them work on their own and have hormone functions. An example is histamine, which is a decarboxylated form of histidine, one amino acid that we've talked about, an important one whose R group can ionize at physiological pH. Histamine is a powerful mediator of allergic reactions, and so it has a hormonal type endocrine response. Also dopamine, it's a modified version of tyrosine, and it's a neurotransmitter that is strongly related to reward signals in the brain. So some modified amino acids can have individual solitary functions and don't need to be included in proteins. So let's talk about primary structure. There's a lot going on this figure here, and we'll come back to this and reference it as we move through the different degrees of structure. For right now, all we're concerned about are these lines. Primary structure is the strand. It's not the shape of the strand, it is the strand. So we're going to emphasize primary structure in this section, but there are close ties to secondary and the other forms of structure, because all of the other forms depend on primary structure. So we'll talk a little bit about the other forms, but we'll emphasize primary. And in the simplest form, primary structure is simply the sequence of amino acids. The sequence of amino acids. So one amino acid interacting with its neighbor. That is primary structure. In practice, that sequence can be changed by modifying some proteins like we just saw. It can be rearranged, and we'll see an example of that when we talk about insulin. Now, primary structure of proteins, like I said, is the sequence, and the sequence is theoretically nearly unlimited. There are 20 possible amino acids to include at any individual position of a protein. And so there are 1.27 times 10 to the 130 possible sequences for a given 100 amino acid protein. And that's a fairly short protein. So a lot of possible sequences. It's more than double the number of observable or atoms in the observable universe. So a lot of possible combinations. And by far it's the most complicated. Proteins are the most complicated structures in the cell. Far more complicated than DNA or mRNA or anything else. Proteins can be the most complicated molecules in the cell. Typically, we limit proteins to 100 to 1,000 amino acids. There are some smaller ones and some bigger ones, but the majority are in that range. In general, when we get too long, there are errors in mRNA sequence, and so synthesis can't be as efficient for longer proteins. And we're limited by the ability to fold the protein as well. So an example that we'll discuss that highlights some characteristics of primary structure is with insulin here. This is bovine insulin. This is the primary structure of bovine insulin. And on the PowerPoint slides that accompany this video, you have a slightly different depiction, which we'll come to in a second. But this is the sequence of amino acids in insulin. Bovine insulin is very similar to human insulin, or insulin in many mammals, and it's made of an A chain, a B chain, and a C chain. The C chain of insulin, surprisingly enough, is removed. In the form as it's displayed here, this sequence is not active. This is printed off, if you will. It's translated from mRNA in this sequence, one long ribbon. And then the red region, the C peptide, is removed. And when that C peptide is removed, insulin is active. So, why does this happen? Why do we have to make this whole protein just to remove half of it? Why not just make the two blue-green sections? The reason is that we need this entire peptide in order for these disulfide bonds to form. After we make this ribbon, the protein will naturally fold according to the influences of all the various side chains in this protein. And some of these disulfide bonds will form. This is required, this entire ribbon is required in order for these bonds to form properly. If we didn't have this red region, and we just synthesized these two blue-green sequences independently, we could link this up one way, the proper way. The A chain could be backwards, it could be upside down, and these sulfide bonds could link to the B chain. So there's no way to know that it would actually form the proper active form that we need. Printing everything out all at once and then removing part of it is a surefire way to know that it will form the proper structure. What you have on the slides is this picture, which is the same depiction of insulin, but without the C peptide. And you can make the C peptide by simply linking the glycine residue, amino acid residue on the A chain, to the alanine residue on the B chain. And what I mean by residue is simply an amino acid that has been incorporated into a peptide. When it's not a free amino acid anymore and it's bound to another amino acid, we commonly refer to them as residues. So this is the active form of insulin. Now it's not as simple as this in the body. Here we have two linear straight chains that are laid out for us to see. But we can tell that there will be some extra folding that happens. And while that's not the focus of this section, it's important to point this out now while we discuss insulin. You can see that the two sulfur side groups on cysteine on the A chain are connected in a disulfide bond here, indicating that this has to fold back on itself somehow. So indeed, even though this is laid out linearly, the side groups of all of these individual amino acids will influence how this protein will structure itself in the cell. So this is one example of a fairly simple amino acid In general, amino acids are limited to around 100 to 1,000 residues in length. Like I said, some are smaller, some are longer, some are very much longer. And the constraints are that proteins need to have at least 40 amino acids to be considered a polypeptide. It seems that 40 amino acids is about the minimum that is required to fold and make a functional active protein. And at most they have usually 1,000 amino acids. There are some exceptions to these rules, but above and below these ranges is where we start to see either non-functional proteins in that they can't fold, or proteins that are too long and there are errors included in them that don't allow them to fold properly and carry out their proper function. Now, the primary sequence of proteins is what underlies all other levels of structure and therefore all levels of function of the protein. The primary structure, the primary sequence of the protein is what contains all of the information and allows proteins to do the work that they're supposed to do in the cell. And so it's important that these sequences stay the same. If they change, then the shape of the proteins will change and the function will also change. And so if we want to keep the same function, we have to keep the same primary sequence. If protein sequences change, that is, if the primary sequence changes, then the function changes, and if that function is better than it used to be, that change will be maintained. It'll be kept around in the cell. If not, usually it will be discarded because that protein will be less functional or sometimes non-functional. This is what we call protein evolution. It's all based on the primary sequence of proteins. So these sequence similarities between different organisms with the same proteins describe how related proteins are, and therefore how related different species are. Now, mutations are happening all the time, and so structures do change gradually over geological timescales, really large timescales. And we can use this information to calculate how related different species are. Generally, if a protein is very important, it'll be inversely proportional to the number of alterations that we observe, the number of changes we observe. So the more important the protein, the fewer alterations that are likely to have happened. That is, the more important the protein, the more similar the proteins are expected to be between different species. And we can see that here with cytochrome C, which is a protein that ferries electrons between complexes in the electron transport chain. In essence, it is the chain of the electron transport chain. Complexes can't move electrons between themselves. Cytochrome C is the thing that does that. It allows the chain to exist. And it is critically important for the electron transport chain to be active to support normal development in the body and provide energy to support processes in the cell. And therefore, cytochrome C is very important in this normal function. It's a very important protein. And so if it's a very important protein, it should be relatively similar across a wide range of organisms. And you can see that by looking at the vertical colors on this graph here. The vertical color similarities indicate similar properties of the amino acids in each of these positions. And if we look across the bottom, we get an overall picture of the number of different amino acids in each of these positions. So across all these 38 species, if we look and see how many keep the same sequence, 38 of them contain one amino acid only. That is, there are 38 positions that are the same. There are absolutely no differences in any of these organisms at 38 positions in cytochrome C. Cytochrome C has only 104 positions. And so that's a large proportion of this protein that is kept identical amongst all of these different organisms. And we say that these positions are highly conserved, that these residues are invariant residues. They don't change. And they don't change because the characteristic of that amino acid is important. Usually the R group here is important. So either it's important to bind to another R group, or maybe it's large and there are space constraints and it forces the protein to fold in a certain way. Whatever it is, the R group here in those positions is important, and so the amino acid doesn't change. Some positions have more than one amino acid, and if there are substitutions that occur, usually the substitutions will be for amino acids that have similar properties. This indicates that amino acids are more or less essential in these areas. When we get to positions where there are more and more amino acids being substituted, it indicates that these positions are less and less important for the overall function and shape of the protein. So, with this being such an important protein, that begs the question, why is the sequence different at all? Why are there any regions that are variable? And this arises from a process called neutral drift, because there are always errors in copying DNA and passing on these sequences between generations. The changes that are bad for this protein are usually discarded, but those that don't decrease the function aren't selected against. They'll stick around. And this is called neutral drift. So any changes that don't change the overall function of the protein, they won't be discarded. So some changes will be allowed to persist. And that's why there is some variation, and it's not kept identical in all of these organisms. And so if we count the number of different amino acids, it gives us an idea of how similar these proteins are. We can calculate a percent similarity. That is the number of different amino acids for every 100 amino acids. And that percent similarity tells us how related the proteins are between different organisms. And we can draw how related these proteins are in what's called phylogenetic tree. This is a depiction of how similar cytochrome C is between different organisms. In this picture, the branch points are theoretical common ancestors, where the sequence would have been the same between two different species. And then the distance between branch points and the number beside the branch indicates the percent similarity. So a branch point here would be 11% different from a branch point here. There would be 11 amino acids that are different per 100 between these two proteins. Now you can see humans and chimpanzees on the same branch. That is that our cytochrome C sequence is identical. And we are most closely, our cytochrome C sequence is most closely related to the rhesus monkey. Now this common ancestor has 6% difference from other mammals on this branch, other mammals in this chain. So cytochrome C is relatively similar amongst all mammals. The common ancestor between us and rhesus monkeys has only 6 amino acids that are different between these other mammals here. If we trace these sequences all the way back and extrapolate, we find this one singular origin point at the end, which is the earliest common ancestor for cytochrome C. It's the point where theoretically cytochrome C had the same sequence, and then we've branched off of this point and developed different organisms with different sequences. So primary structure, if we summarize quickly, is the sequence of amino acids, and it underpins all other forms of structure. Changing the DNA changes the mRNA. It might change the code for which we make the primary structure, and will change the sequence of proteins slightly over time.